The purpose of transcription is to unzip DNA into two template strands so that a complementary strand of RNA can be made. There are 3 steps in transcription: initiation, elongation, and termination. In initiation, RNA polymerase binds to the promoter region of a DNA double helix and unzips the 2 strands. In elongation, RNA polymerase reads the template DNA strand and builds a complementary RNA strand. In termination, once RNA polymerase reaches a stop sequence on the DNA template strand, it stops building mRNA and pops off the DNA strand. Before translation can occur in eukaryotes, the mRNA strand must go through a pre-translation phase, also known as RNA processing. There, a 5' end cap and 1 poly A tail is added to the 3' end. Be sure to remember that the pre-mRNA is released soon after the polyadenylation signal is transcribed. Small nuclear ribonucleoproteins, also known as snRNPs, and other proteins form a molecular compound known as a spliceosome. This spliceosome cuts out the intervening sequences, or introns, leaving the exons, which will later be expressed in translation. Once the mRNA strand has gone through pre-translation, it leaves the nucleus and goes into the cytoplasm, where it will be read by ribosomes in translation. Ribosomes are made up of one large subunit and a small subunit. There are 3 sites on a ribosome: the A site, the P site, and the E site. The A site holds the tRNA, carrying the next amino acid to be added to the polypeptide chain. The P site holds the tRNA attached to the growing polypeptide. The E site is where the discharged tRNA leaves. A large ribosomal subunit will connect to the small subunit, and the initiator tRNA is in the P site. The A site is available to the tRNA bearing the next amino acid. In the elongation cycle of translation, an incoming tRNA base pairs with the complementary mRNA codon in the A site. The amino acid on the tRNA at the P site forms a peptide bond with the amino acid at the tRNA that's at the A site, adding to the growing polypeptide chain. Once the tRNA at the P site has lost its amino acid, it moves over to the E site, where it will be discharged from the ribosome, and the tRNA at the A site will move over to the P site in order for the process to repeat again. When a ribosome reaches a stop codon on the mRNA strand, the A site of the ribosome accepts a release factor. During this time, the tRNA in the P site is also released, as is the polypeptide chain. This now free polypeptide goes on to become a functional protein, and the 2 ribosomal subunits dissociate. In eukaryotic cells, transcription occurs in the nucleus of the cell, while translation occurs in the cytoplasm. In prokaryotic cells, transcription and translation both occur in the cytoplasm. Prokaryotic cells also have no pre-translation